Share. Well, now.
That's beautiful. It's
All
perfect.
right, finally, we got there. All right, so let's dive right in. So, as I mentioned, my lab's really interested in diabetes. Type 1 and type 2 diabetes affects over 35 million Americans, which is over 10% of our population, and accounts for around $250 billion in medical costs and lost wages. So it's a tremendous problem. It creates many complications, just a few of which are listed here. So for example, patients with diabetes have about a 1.5 times greater increase of stroke, twice as much chances of being affected by hypertension, kidney failure, heart attack, 20 times increased likelihood of having nerve damage, 5 times more likely to undergo a limb amputation, and 25 times more likely to become blind as a result of their diabetes. And so my lab studies microvascular biology, and one of the striking things about all of these complications is that you can really bring them down to the level of the microcirculation as a contributing factor. And so that's what my lab studies, is the changes that are experienced in the microcirculatory system as a result of diabetes. And today we're going to start out by talking about blindness, because there isn't another tissue where the manifestation of this disease is so vivid. You don't have to be an ophthalmologist or a retinal surgeon to appreciate the fact that when you take a standard picture of the back of the eye, this is just the fundus imaging that you would experience if you went in for your annual eye checkup, that the vasculature is completely disorganized in a diabetic patient's retina as compared to a healthy patient, which is shown on the left. So what a diabetic patient who has a retina that looks like this, with this disorganization of the vasculature, would see is something like here. They're going to experience blurred vision. They're going to experience parts of their vision field that are either completely blacked out or have white spots. And it's obviously very disorienting, very troubling. And it's going to ultimately lead, if untreated, to blindness, because these aberrations in the microcirculation will eventually turn into leaky and then fibrotic tissue, which peels the back of the retina off the back of the eye, the retina off the back of the eye, and so ultimately the patient will become blind. So it's a terrible, devastating disease, progresses over the lifetime of the patient. And when you look at the cell biology that's associated with these changes, which you can see here, it's important to really look at 2 cell types in particular, and these are the cells that our lab has spent almost 16 years studying, and that is the pericyte and the endothelial cell. So let's zoom in to these retinas, and you can see endothelial cells labeled in blue and pericytes labeled in green. Now, pericytes are not parasites. They're supposed to be there. They're the cells that wrap around the endothelial cells and provide support. And you can see in the diabetic retina, they look morphologically quite different. If we zoom in even a little bit further, you can see that, in fact, whereas the healthy retina has pericytes that are nicely juxtaposed along the endothelial cells, they're hugging on tightly and, as I said, providing support to those capillary endothelial cells. The diabetic retina, this pericyte, is exhibiting a morphology that we've recently characterized and published that is quite unusual, I would say. We call them pericyte bridges, but they've essentially peeled off of the capillary endothelium, and they're kind of hanging out in outer space, in the interstitial space, but they're still connected to the capillaries. And so we think that there's some really intriguing and important alterations of the vasculature that results from these pericytes kind of behaving differently in diabetes. So we've quantified this behavior in mouse models of diabetes, and we've shown with a lineage tracing mouse where we have basically all of the pericytes are fluorescently labeled, and we can track them over time. We've shown that when we create diabetes in these mice by injecting streptozotocin, which is a toxin that kills the beta cells in their pancreas and makes them hyperglycemic, there's essentially a doubling of this behavior. So on the left, we see these blue circles representing data from mice that are non-diabetic, and in the red diamonds, we see data from mice that are diabetic, and the fraction of these pericytes almost doubles when these mice have been treated with diabetes. And so we think there's something important there that might map to the more macroscopic observations that we've had, which is that, and this is the cornea now, you can see a diabetic mouse's cornea that has undergone a burn to induce angiogenesis, a chemical burn applied to the top of the cornea. You see these very obvious, without even a microscope, you can see these hemorrhages, which result in response to that wound healing. And that is, again, abnormalities in vascular growth. So that's in stark contrast to the healthy cornea, the healthy mouse cornea, where we see angiogenesis proceeding as we would expect it, where the vessels are nicely aligned and extended, and they're not forming these hemorrhages that we see in the diabetic. So we think there's some relationship there between those pericytes behaving in that interesting bridging way in the diabetic eye and this more macroscopic dysfunction that we can see in the cornea when it comes to making these hemorrhages. Furthermore, when we have studied the permeability of these same vessels in response to a wound, we see that a healthy vasculature is nicely perfused when we inject a red fluorophore. You can see all of the red dye essentially remains within the vessels, and these vessels are nicely covered with green pericytes. However, in a mouse that exhibits diabetic-like features in terms of a diminishment and abnormalities in pericytes, shown in green, you can see when we inject the same red dye that the vasculature is extremely leaky, again representing sort of a more tissue level response to what we think ultimately is stemming from the fact that the pericytes are pulling off of vessels in the diabetic eye. So this was also kind of recently published. Now, one of the most fascinating things about this, which has really led us to be motivated to turn back to our models, which I'm going to talk about next, is that this phenomenon seems to be entirely reversible. So with insulin treatment. So we've done an experiment where we've taken 3 groups of mice. One group has received just a vehicle, and it's a healthy mouse. Another group of mice received that streptozoosin toxin that I mentioned that turns a healthy mouse into a hyperglycemic or a diabetic mouse model. And the third group of mice received that in addition to an insulin implant 7 days after the STZ injection. And so they essentially received insulin therapy for a week. And what's interesting is that after 7 days, we see what I showed you before, which is that there's an increase in the fraction of those pericytes becoming bridges and extending off of the capillaries. But that is returned almost to normal levels by 14 days when we have implanted the insulin pumps into these mice. So it suggests that this happens very quickly, but also you can reverse it over a very short time scale. We've repeated the same experiment using a longer time scale, where we've injected or inserted the insulin implant at 7 weeks and left it in the mouse for another 7 weeks. And you can see the same thing happens. We get a return to normal levels in terms of the fraction of pericytes that are behaving like they should. So this is all very encouraging. And if in fact these behaviors map to those higher level morphological abnormalities I talked about in the cornea, then it says that there's an opportunity here for us to think about how we might deliver some therapies that could target pericytes and prevent them from doing this, which then would hopefully prevent some of those pathological abnormalities we see in the microcirculation. So it's also encouraging that we know from other studies that by taking videos of pericytes interacting with endothelial cells, that they're very dynamic. So this is a video of mouse embryo where the pericytes are labeled in red and the capillary endothelial cells are in green. And this white arrow is pointing you to a pericyte of interest. If you can keep your eye on that, you'll see that in fact it is really interesting behavior that this pericyte exhibits. It actually gets up and it walks off of the vessel it was previously situated on and comes over to this other vessel, which ends up being a capillary sprout, and kind of grabs onto the end of it. We see a similar phenomenon in terms of pericytes being dynamic in the adult mouse cornea. Here I want you to fix your eyes on this little red pericyte, and if you watch over time, it also decides to get up and kind of extend a protrusion and then ultimately walks off of the vessel, forming, like I said before, what we think are these pericyte bridges that connect across capillaries. But it's very unusual for normal parasites, so to speak. So we know that this can happen in healthy tissue. We think it's accentuated in the diabetic, and this leads us to, as I said, really want to understand how parasites contribute to complications of diabetes, so that if we can figure that out, we can target them for therapy. So in our lab, we combine the experiments that you saw, which involve dynamic imaging of real life tissues, with computational modeling, specifically agent-based modeling, so that we can simulate endothelial cells and pericytes and other cells of interest at the individual level, and then observe patterns or features or changes that are emergent and consequences of the individual actions at the tissue level, or in our case, the whole network of the vasculature. So it's important for our models to really explicitly represent heterogeneity and dynamics. And as I mentioned, we like to use agent-based models for doing that, inspired, of course, by the prolific application of agent-based modeling in ecology. I like to use this example for anyone who hasn't encountered agent-based modeling. Basically, the idea is represented in these Canadian geese, which, as we all know, when they're assembled together, tend to fly in a V-shaped flying formation, which you can see right there in the video. So from the modeling standpoint, we can represent these individual geese, or in our case, cells, as autonomous agents. They are able to follow a rule set which tells them how to behave given certain individual conditions they might encounter. The rule set will dictate what behaviors that they will exhibit as their environment changes. And then the end result of those individual behaviors, as I said, is that aggregate patterns emerge. And those patterns are at the higher level of scale, in our case, tissues like the microcirculation. So representing individual cells as agents is actually pretty straightforward. We've been doing it for a number of years now. We usually start with an image or a picture of our tissue, and usually these images come to us in two-dimensional forms. So almost all of our models to date have also been two-dimensional. And we, in this case, have traced all of the boundaries, the cell membranes of each of these cells, and we incorporate them directly into our modeling software platform. We've used NetLogo and Repast, and we're experimenting with some other platforms, and there's some wonderful new platforms that have recently been published, and I can refer you to those later if you're interested. But we basically ascribe an individual agent for each cell. We endow it with certain characteristics, the ability to exhibit behaviors that we see in biology, such as proliferation and migration, differentiation, apoptosis, or cell death. And we give it a rule set that tells each agent when to exhibit those behaviors. And then we run the simulation, and we can, watch the dynamics the same way that we can watch the dynamics in the videos that I showed you earlier. So we have made agent-based models to study angiogenesis, and we have more recently made agent-based models to understand the role of pericytes in angiogenesis. And so this is showing a model of retinal development, retinal vasculature development. This is not a diabetic mouse model yet, but it's a model of development. And we have incorporated 3 different cell types. We've incorporated astrocytes in green. The astrocytes are critically important during angiogenesis in the developing mouse eye because they secrete boatloads of one of the most important pro-angiogenic proteins that was identified back in the 80s, called VEGF, or vascular endothelial growth factor. These models also incorporate, obviously, pericytes as well as endothelial cells as the other 2 key cell types. Each of these cells is able to respond to its localized environment. So it's responding to concentrations of VEGF, it's responding to the proximity of neighboring cells, and there is a requirement for those cells to stay connected to one another and to signal through one another, to signal to one another through the Notch DLL4 signaling system, which is a membrane-bound ligand that binds to a receptor on an adjacent cell. And there is the presence of the pericyte, which serves as an obstruction to these interactions and also changes the shape of the gradients of VEGF. So all of those, essentially what I just told you, are rules that must be incorporated into our model, and they're incorporated just with very straightforward mathematical equations, algebraic equations, or ordinary differential equations that will govern the rate of change of certain protein expression as a function of binding of a ligand to a receptor, for example, as you can see in these equations, where we're dictating the amount of VEGF receptor protein production being dependent on the notch activation, and the notch activation in turn being dependent on the amount of ligand that activates it in the adjacent cell. So our models will typically have 50 to 100 rules, depending on how complex they are. And the output of these rules is nicely captured in movies. So we can actually see the agents responding to changes in this growth factor concentration. They're migrating and proliferating. They're moving up gradients. They're holding on to one another and pushing apart one another. And in the end, we can map changes over time to what we observe experimentally and see whether or not our agents are valid under those particular conditions, which in this case is the release of VEGF from astrocytes in a developing mouse retina. So we've done that, and we did it for 3 different cases because we're so interested in the importance of pericytes and what role they might be playing when they're interacting with endothelial cells. And it turns out that when we incorporate endothelial cells and pericytes into our simulation, we're better able to predict the total vascular length density that we measure in actual retinas that were harvested from postnatal day 3 mice. That data is represented in the gray bar. And we see when we only simulate endothelial cells, there is an increase, almost a doubling of vascular length density. And then when we simulate sparse endothelial cells, or just a reduction in the number of endothelial cells, we can bring the length density back down to more normal levels, but it's not quite at the level that we see when we're able to, when we have the pericytes included in our simulation. So this is a little bit of validation that our model is relevant for the case of endothelial cells and pericytes. But what's more interesting and was more helpful for us to sort of interpret the consequences of that pericyte bridging behavior in diabetes is what I'm about to show you. So this is now information from our model that we could not glean in any other way doing experimental techniques, basically showing protein expression of multiple proteins at the individual cell level. Easy to predict and calculate with an agent-based model, very challenging to actually measure this experimentally from a harvested mouse retina. So what we're looking at in the top 2 panels is the situation where we have endothelial cells and pericytes present. The blue cells here you can see that are kind of colored according to this color scale are the endothelial cells. These gray outlines are representing the locations of the pericytes. And what's interesting is if we look at the notch signal in one cell, say this one here that the arrow is pointing to, it's relatively low, but in that same cell, the ligand for notch, which is DLL4, is relatively high. Likewise, if we look at 2 adjacent cells, we can see that the notch ligand in this cell right here with the other arrow is relatively low, but its adjacent cell has notch that's relatively high. So there's this sort of salt and pepper patterning of the notch and DLL4 signal that is representative of what we think happens in vivo and is important for controlling which of these cells gets to sprout and which of these cells is inhibited from sprouting so that we get a more normal network. Now, when we simulate the removal of pericytes and only have the endothelial cells present, you can see the expression patterns of notch and DLL4 kind of go haywire. So in the same cell, we'll have high expression of both notch and DLL4, and also in adjacent cells, we'll have very high expression of the same protein, which again is kind of unusual, uncharacteristic, and leads to that hyperangiogenesis that we have predicted. So this is kind of interesting and makes us think a little more deeply about the role of the pericyte and maybe its essentiality for keeping cells apart from one another, keeping the endothelial cells apart from one another so that they can sufficiently organize their notch and DLL4 signaling to manifest in an emergent pattern, a higher level tissue pattern, which is a more organized network and not what we see in the diabetic eye where the blood vessels are sprouting like crazy and kind of going haywire. So these types of simulations led us to want to explore possible treatments that could restore the normal distribution of signaling in endothelial cells by providing a tissue with additional pericytes or parasite progenitors. So we've done a number of studies now, again, in mouse models, where we have used parasite progenitors harvested from a tissue that is highly vascularized, which is our adipose tissue, and also very easy to obtain through liposuction or lipectomy. It's also a wonderful source of parasite progenitor cells, which are very easy to harvest, expand, and culture, and then re-inject into your tissue of interest where you want to provide additional parasites to a situation where vascular abnormalities are present, such as a diabetic eye. Now, as I mentioned, we've put these cells into not just eyeballs, we've injected them into chronic diabetic wounds, we've injected them into skeletal muscle of diabetic mice, and we always see the same thing, which is that these progenitor cells like to occupy locations that are very typical of parasites. So on the left, you can see one of our progenitor cells that we have labeled, and it on its own just was able to migrate to an endothelial cell in a tissue. And this happens to be a rat mesenteric capillary, but it showed up in almost the exact same location where we see the native parasites. And so this suggests, again, we have ourselves a source of parasites that we can inject as a stem cell therapy to ideally restore the vasculature back to a more normal pattern by reorganizing the cell-cell interactions between endothelial cells. So at the tissue level, this actually bears out quite nicely in a number of mouse models that we've published over the years. We see on the left mouse retina that has been injected with saline or vehicle control. And in the very same mouse, we can see the right retina that has been injected with these parasite progenitor cells derived from, in this case, mouse adipose tissue. Because we didn't want to have to worry with confounding effects of the immune system rejecting human cells. So in this case, we've injected mouse cells. And what we see is that there is a return in vascular density to more normal levels as a result of having those parasite progenitor cells in place. And when we zoom in and track them and say, did they actually go where we expected them to? We do see green parasite progenitor cells. These were pre-labeled with GFP. They are able to associate with the vasculature. Now you'll see there's a little bit of disorganization, actually. They're kind of, in some cases, a little bit maybe too abundant. And so that actually was something that we kept in our back of our mind. And it did kind of, cause us to think maybe they're doing more than just associating with the vasculature. Maybe they have other roles that they're playing in that eye. So it turns out that suspicion was corroborated by a study, a human study that I'm going to talk about in 1 second. I just want to pause here briefly. I don't know how we're doing questions, Heiko, but this would be a good place for me to kind of pause and summarize. The results I've shown you so far is that through the experiments and the modeling, we have a better appreciation for how parasites might be contributing to complications of diabetes, specifically by regulating this endothelial cell signaling via the Notch DLL4 axis. And then following up on that by changing the number of parasites in a tissue, in a diabetic tissue, to alter that cell signaling and return a vascular pattern to normal. Through the use of parasite progenitors. So there's a good pause if we want to do questions. I don't know what your protocol is for this. I can keep going and take questions at the end. Would that be preferred?
Yeah, let's do that.
Okay, sounds good. Okay, so I warned you about these human studies, and actually, truthfully, this wasn't even a human study. It was more a situation, I would say, where 3 patients went into a stem cell clinic, and they received, this is back in 2016, they received injections of adipose-derived progenitor cells, very similar to the ones that we had injected into our mice with diabetes. Now these patients, they were not diabetic, they were suffering from age-related macular degeneration, and they had visual acuities that ranged between 20/80 and 20/200. So they were not, they had pretty poor vision, but they were not blind, they did have some vision. And these 3 patients were injected with these cells into both eyes, so bilaterally, both eyes received injections. And within a matter of months, their vision, what they had left, was markedly diminished to the point where all of the patients became blind in all of their eyes. And ultimately, this was attributed to fibrosis, scarring, and retinal detachment. So this is a case where obviously the exciting results that we had obtained in our mice did not at all translate to a benefit. In fact, it was worse than that. It created severe impairments for these patients. And so this report, along with some other research that we've since done in the lab, has made us really focus on what these pericytes are doing at a deeper level and whether or not they might actually be responding to fibrotic and inflammatory cues, which are rampant in diabetic tissues, especially the eye, in ways that are similar to fibroblasts as they undergo myofibroblast transitions. And so sort of the provocative hypothesis here for the eye is that these pericytes, which we know when they're behaving properly, help stabilize endothelial cells and regulate vascular permeability and angiogenesis, in the situation of a diabetic where there's fibrotic and inflammatory cues, they might in fact be differentiating into myofibroblasts and contributing directly to scar formation. And so I want to show you some of the biological evidence we have for this, and then how we're using our models to dig a little deeper into this potentially, concerning but possibly helpful avenue if we can flip it into a therapy that we could use to target this process. So we have a mouse model. It's an in vivo injury model where we essentially, as I mentioned, burn the cornea, creates massive inflammation and ultimately fibrosis. We see TGF-beta levels increase from around 150 picograms per mil up to almost 800 picograms per mil. That's in the matter of 7 days. We also see various chemokines, CXCL10, for example, increase tremendously. It's a pretty bad burn, and the response of the eye is inflammation and fibrosis. And you're kind of looking at 2 of the hallmark factors that represent that. Now, in that same eye, if we go and do fluorescence imaging on the cells, specifically the pericytes, which, as I mentioned, we have a mouse that expresses fluorescent marker, in this case it's TD tomato, in all of the pericytes and their progeny, we see these cells do things that we never see in healthy tissues, which is upregulating expression of MYH11, myosin heavy chain, upregulating expression of submuscle alpha actin, upregulating expression of collagen 3, and ultimately exhibiting these morphologies that don't look anything like the cute little pericytes perched on the edge of the capillaries, but much more like what any expert who's in the field of fibrosis would recognize as a very angry fibroblast. And so we see that time and time again, not just in the eye, but in other tissues such as the lung, where pericytes that are exposed to these factors, TGF-beta and chemokines associated with inflammation, under the right conditions can really start looking like a myofibroblast and contributing to scar formation in that tissue. We have also done studies in vitro where we'll culture these pericytes in TGF-beta and then look for expression of various proteins such as IL-6, inflammation marker, VEGF, MCP1, all of these go up significantly when we treat these pericytes in a dish with TGF-beta. So again, underscoring the point that these pericytes might have a dark side. They might be able to undergo fibrosis, contribute or undergo transition to a fibrotic cell type and contribute to fibrosis in that tissue. We've also done analysis of this transcriptome, looking at bulk RNA-seq from lungs that have been inflamed. We would love to do RNA-seq on retinas, but the fact of the matter is in the mouse, you don't get very much material, so we haven't mastered that yet. But when we stimulate pericytes and endothelial cells in the lung to be highly inflamed using a bleomycin model, which is basically a toxin that you inject into the lungs, and it creates rampant inflammation. after a matter of weeks, 1 week or 2 weeks, we see marked changes in the transcriptome of pericytes and endothelial cells, but especially when we look at the genes associated with collagen and ECM, or extracellular matrix regulation. So again, in addition to protein, there are changes at the RNA level as well, which map to things that look like fibroblast or myofibroblast transition in these sweet little pericytes, which are supposed to just hang out on the endothelial cells. So we've taken that information and turned back to the modeling, because obviously there's a lot there in terms of the integration of signals that these cells are doing, as well as the behaviors that they're eliciting. So in collaboration with Jeff Saucerman here at UVA and with Jeff Holmes, who's now at University of Alabama, we have developed a multi-scale model that couples a intracellular logic-based signaling network model developed by the Saucerman lab a few years ago that incorporates 10 signaling pathways and 11 biochemical stimuli. We've incorporated that directly into an agent-based model where we've basically copy-pasted this little representation of a fibroblast 100 times, and we've placed it on a two-dimensional landscape of factors with different increasing gradients. So from bottom to top, we have a landscape of increasing TGF-beta concentration, and from left to right, we have a landscape where we have increasing inflammatory cue. Again, keeping in mind that in the previous slide, I just showed you that our pericytes are highly responsive to stimulation by TGF-beta and IL-6 in particular. So when we put these cells down in this landscape and then predict over time with the agent-based model coupled to the network model how collagen would change, you can see this sort of map of increasing collagen over time. And it makes sense in this case that we would see that the representation of fibroblasts that are residing in the top left corner, where there's a high concentration of TGF-beta, would have a very high expression of collagen. That makes a lot of sense. Likewise, in the bottom left, low TGF-beta would cause low collagen expression. And we've plotted this over time and for different configurations and different concentrations. And that data is summarized here in this grayscale. So again, we have the steady state collagen area fraction that is produced by these simulated fibroblasts with that network model functioning. And we've shown that if you can just focus on the four corners, the control case, which is virtually zero inflammatory cue or fibrotic cue, we get virtually no expression in collagen, as you might expect. When we move over to the case of TGF-beta by itself, the high concentration, this light purple bar, it's represented here in the second bar. This is the result of the collagen being deposited by the fibroblast that lives in that neighborhood. And then you can see over here, the third bar represents high concentrations of the inflammatory cues, IL-1 beta in particular, and we see that there is, again, a little bit of collagen being produced by that cell, but not very much. But when we combine the two, the cell that lives up here in the top right quadrant, we see that there is more collagen being produced, but not nearly as much as that's being produced by the cell that lives in just the high concentration of TGF-beta alone. So we wanted to kind of validate that result, that the expression or the presence of an inflammatory factor attenuates the response to TGF-beta in terms of collagen production. And we did so by putting cells in dishes, fibroblasts in dishes, and they're labeled here, blue nuclei. And you can see when we stained for pro-collagen 1, the qualitative pattern that we predicted by the agent-based model coupled to the intracellular signaling model is quite consistent. So control has some baseline expression. It's markedly increased for the cells that are cultured in TGF-beta alone. We can bring that down by co-culturing them with not just TGF-beta, by culturing them with TGF-beta in combination with the inflammatory cytokine IL-1 beta. So in this way, our model prediction is kind of nicely validated by this very simple experiment. We're just exposing these different growth factors and chemokines in combination and alone to these cells. So moving on from that, I guess, you know, we've kind of shown that this approach is able to predict how the environment, specifically collagen in the environment, is changed by the presence of these different cells. But the flip side of that, as we know, is that cells are sensing their environment. So we can use the same simulation framework to predict how the environment changes the cells over time. And so to represent how we do that, we've simulated two different fibroblasts. So the top row of data that you're looking at is from, we'll just call it cell number 1 or fibroblast number 1. And the bottom is cell number 2. It's the same data, just reported for a different cell that we've simulated. So on the far left graphs, what we're looking at is basically the time course trajectory of where these cells went in this landscape when they were allowed to migrate randomly. And so blue represents the earliest time point and red represents the latest time point. So this is sort of the journey of the cell. Started over here in the land of low inflammation, medium fibrosis, meandered its way down towards the inflammatory direction and over to ultimately where it finished its journey, which was in the land of low fibrosis, high inflammation. And when we look at the gene expression predicted by the model, we see that it experienced, because of its traverse through this landscape, it experienced at the beginning a medium amount of TGF-beta, gradually increased and then decreased and kind of remained steady. But the amount of IL-6 it experienced was very low to begin with, as I mentioned, and then gradually went up. And commensurate with those input signals, the output signals were also kind of interesting. So we see collagen mRNA pretty much stays at a low expression level for the whole time. So this particular fibroblast going through that trajectory doesn't really express much collagen. However, it does express an increasing amount, a gradually increasing amount of matrix metalloprotease, which is, of course, going to be a factor that would degrade collagen. So that's the story of this particular cell as it goes through its journey. And if we want to see a video of what happened in terms of each of the gene nodes that were represented for those signaling pathways, we can see that play here. Now that is in contrast to another little cell who took quite a different journey. It started out over here in the land of higher inflammation, meandered around, and then ultimately worked its way up to the region where fibrotic cues were the highest levels of TGF-beta, but low inflammation. And when you look at the expression of collagen, it was definitely more than the first cell. And also it had an earlier trigger of MMP expression over time. And then its video is shown here. There's not a ton of information you can see at this scale, but when you dig into each of these individual genes, you can really appreciate the connectivity of these factors to one another and how a subtle change in one might propagate to a big change in another. And certainly the signals that are being input to the cell as it makes its traverse across this landscape of gradients are affecting in different ways the output that the cells will have. So this is, I think, a useful tool now for predicting how cells sense their environment and what effects that will have on them. But in this case, we're still only representing cells as individual agents that basically have the same rule sets. They have the same underpinning network. The only difference in these cells is where they happen to be when and what signals that they experienced over that time course. But what we know now, as we dig more deeply into fibrosis and that process and fibroblasts and pericytes as well, is that no two cells are created equal. In fact, there are subpopulations of these cells that are really important to take into consideration. So in collaboration with Tom Barker, who's a colleague of mine at UVA, he's a lung fibrosis expert. He has developed a data set where he used single cell RNA-seq, and he applied that tool to analyze fibroblasts that had been exposed to various chemokines in combination. And from doing this sort of high throughput single cell analysis, he was able to derive, just using standard clustering algorithms, that there were 7 different populations, or I should say subpopulations of fibroblasts that had markedly different responses to these chemokines and growth factors. And that data is represented here in this dot plot. We have in particular 2 populations that we're really interested in. There's a population 0, as he terms it, that is sort of representative of what you consider kind of the standard fibroblast. And then there's a different population called population 5, it clustered together under population 5, and it has quite a different response from population 0 to some of the chemokines that we're most interested in. And so we've now incorporated the specifics of those populations into that intracellular signaling network model. And we're in the process of putting that information back into the agent-based model so we can really understand how these different subpopulations of fibroblasts and ultimately, eventually, pericytes are working together to both sense their environment and change their environment for other cells that might follow behind, such as the endothelial cells, which are the ones, as we know, contributing to that vasculopathy that we see in the retina of the diabetic patient. So just to sum up, again, we've really tried hard in our lab to do a tight integration of experiments with modeling, specifically agent-based modeling, but also more recently coupling those models with intracellular signaling network models, and ultimately ask or answer the questions that I mentioned are really the driving force behind our lab, which is how do these fascinating little cells contribute to the complications of diabetes, and are they a suitable target? So I think thanks to the recent efforts, appreciating more deeply that these pericytes can exhibit myofibroblast tendencies, and also, as terrifying as that might be, it might be an additional opportunity for us to target them in terms of returning their phenotype, their morphology back to normal, where they reside happily on the outside of their capillaries and remain in a support role as opposed to a fibrotic role. My last slide before my acknowledgements, it's actually starting my acknowledgements, is that I've benefited greatly from many collaborations at UVA and other places, but I just wanted to acknowledge some of our work in agent-based modeling in other tissues and organs. So as I mentioned, we've been collaborating with Jeff Saucerman and Jeff Holmes using agent-based models to study cardiac remodeling, particularly following infarction or heart attack. We've collaborated with Jenny Munson, who was at UVA. She is now at Virginia Tech to model cancer, as well as other people like Matt Lazzara and Kevin Janes, Jason Papin to model infection, collaborated with Sylvia Blunker and George Christ to develop agent-based models of skeletal muscle adaptation, both in diseases such as muscular dystrophy, as well as injury and regeneration, such as volumetric muscle loss. And then last but not least, I mentioned my collaborator, Tom, and his work in the lung, and we've developed agent-based models of the lung as well. And last but not least, I've even been successful at converting the chair of cell biology at UVA, Doug DeSimone, into an agent-based modeler. We have a few publications where we've used the agent-based modeling technique to study embryogenesis and fundamental questions about developmental biology that we're interested in. So with that, I also need to thank all of my students who've contributed to this work. Their names and their publications were flashed up on the screen when I showed their data, but specifically to acknowledge the contributions of Bruce Corliss, who was really the one who characterized in exhaustive detail the formation of those pericyte bridges in diabetes, as well as Clifton Ray, who did a tremendous amount of work looking at pericytes and differentiation of pericytes into fibroblasts in the retina. Other work by Molly, the cornea work you saw was done by Molly. These are all recent graduates, recent PhD graduates from my lab, as well as other students who contributed substantially to the agent-based modeling efforts, Kyle Martin and Joe Walpole. And then my current students who are carrying these projects forward, in particular, Riley Hannon, who's working in the lung, and Julie Leonard-Duke, who's also working in collaboration with him to establish the framework for integrating single-cell RNA-seq data into agent-based models. And last but not least, Michaela Ricard, who really achieved that coupling between the intracellular signaling network and the agent-based model, as well as many collaborators, all of whom are listed here on the right. I specifically like to point out Denise Kirshner, who, as we know, is the past president, and my conversations with her and my relationship has really developed my thinking over the years about agent-based modeling, and I've benefited tremendously from those conversations. So I wanted to thank her in particular. And again, thanks to all of you for your attention. Apologies for the rough start with the Zoom screens, but thank you for being here. And I would be more than happy to entertain any questions now or offline later in a chat or through Slack or all those fun tools that are being integrated into this meeting.
Well, thank you, Shayn, for that really great talk. So if people want to ask questions, please use the Q&A function. We have two already. So the first question, it says, in diabetes, we observe an increase in pericytes and also an increase in decoupling of PC and EC. Do these PCs have any other functions?
Yeah, so I'll take the last part first because it's easy. The answer is probably. And we know from other studies, not necessarily in diabetes, but we know that they play a role in trafficking of leukocytes and inflammatory cells. So they're kind of gatekeepers for extravasation in some ways of other cells of the immune system, which is a really important role. In certain tissues, the brain in particular, we know that they play a contractile role and can help regulate blood flow. So those are definitely additional roles that have not been explored to my knowledge in the setting of diabetes, but could certainly be, you know, more than just the story of endothelial cell support and regulating angiogenesis. The other part, what was the very first part? Oh yes, so interestingly, we showed, I think the question said we see an increase in pericytes. Actually, in that STZ model early on, we don't see any changes in pericyte number. We actually see the same number of pericytes. It's just that a proportion of them, twice as many to be exact, are converting to this other morphology or phenotype, if you will. They're forming bridges, but we don't see a change in total numbers, which was interesting to us. I think, you know, diabetes and angiogenesis in diabetes is really hard to study because it depends where in the disease timeline you're peeking into the vasculature. Sometimes you will see that there's increased pericytes because it might be a region and a time point where angiogenesis is going wild and the pericytes are trying to keep up. But in other times in diabetes, you might actually see regression, and then you might see fewer pericytes. So the whole idea of looking at pericytes in tissues at snapshots in time with diabetes, which is basically what we're limited to, especially in the eye, because it's so darn hard to image the back of the eye in a mouse because the lens is gigantic. The optics are really challenging to do single cell imaging. But in lieu of that, we have snapshots, and those snapshots in time are just not sufficient. So that's another foundational motivator for why we want to do agent-based modeling, is that we can make these movies with our models. We can track cells in time. So those are really provocative questions you've asked, and I don't think we have the answers, but I'm hoping that our modeling of these cells can really kind of fill in the holes with the limitations of experiments.
Okay, great. There are several questions rolling in about the agent-based models. One is how long it takes to run a single simulation, and then there are several people that have asked about elaborating a little bit on the single cell transcriptomics and how you couple that into the agent-based modeling.
Yeah, so the timing question is, it of course depends on how big our model is, how many cells are involved, how much simulated time we are running them for, how many rules they're responding to. So each individual cell, if it has to respond to 2 rules, that will be obviously a much quicker simulation than if it has to respond to 100 different rules. So right now, for most of the models you saw, we can run them in a matter of hours, but that's just for one set of parameters. I didn't mention this, but in agent-based modeling, there's a lot of stochasticity. And so they're not deterministic models. You will get a different output every single time you run the model because there's rules that incorporate probability. So it means we'll have to run our simulation not just once, but maybe 10 times or 50 times to sample that space. So that also increases the amount of hours that these models are churning. We do use our cluster here at UVA. That definitely accelerates things to have things going in parallel tremendously. The other thing that adds time to our simulations is when we're doing parameter sweeps, which we frequently do. It's not enough to just build your model using one set of parameters. You need to fiddle and futz with all the parameters and many of them simultaneously to really explore the influence and the sensitivities in your parameter space. So that also adds a lot of time. So I would say the longest run we ever done has been probably a week and a half. That was brutal, and it was because we were doing a very thorough parameter sweep. But if we just wanted to run the simulation one time with one parameter set, I could probably do that on my laptop and show you that in about 5 minutes. And that's for the parasite endothelial movie that you saw. Now, in terms of the single cell RNA-seq stuff, so this is brand new for us. None of that is published. We're still trying to figure it out, honestly. But basically what we're doing is we're sorting the genes based on both the expression level, relative expression level, but also on the difference in expression between the different subpopulations. And so from that, we're really trying to figure out what makes population 0 different from population 5, and how can we capture that and incorporate that into this signaling model that we have of the fibroblasts. And what we're finding is that in doing that, we also have to expand that signaling network. So the 10 equations or 10 inputs and 11 signaling pathways is not sufficient right now for capturing all the differences that we see in these subpopulations. So it's really a work in progress. Step number 1 is figuring out what signaling networks need to be included. And step 2 is figuring out how to color them pink for population 0 or purple for population 5. So right now we're doing it manually. We're getting ready to apply a little more nuanced machine learning to that process, but it's still a work in progress. And if anyone's interested in that, I would absolutely love to chat further because I think, you know, the abundance of single cell RNA-seq data is only increasing, and our models can be made so much more powerful if we can incorporate that into specifying these individual cell subpopulations using it. So that's a work in progress, and I'd love to chat further and work together with people on it.
Yeah, I think that's a really complex problem. So thanks for answering that. So we have a couple biological questions. One asks, have you considered trying to model the blood flow through the vasculature and the effect of flow on some of the cells, for example, shear wall stress?
Yeah, that is an awesome question. We've dabbled a little bit, but we haven't combined in any kind of productive way computational fluid dynamics modeling with these agent-based models. It's absolutely something that needs to be done. Other people are doing it, and I very much admire and respect their work because I have a very strong appreciation for the fact that endothelial cells respond to shear stress. We've even published papers where we've imaged and measured blood flow and looked at gene expression changes in endothelial cells and in the capillaries, and it is real and it's important, and we should be including that in our models because it's certainly affecting the endothelial cells, probably by association affecting what their pericytes are doing as well. It's an awesome question, and it's something else I would love to work with somebody on, and there's also other work out there that's doing that and doing that very nicely.
Okay, so speaking of their cells, somebody has a question about, says, are the pericytes the first cells to show abnormalities after STZ treatment? Or do they react to problems with the endothelial cells in the vasculature?
I would love to know the answer to that. So we say it's the first thing that happens because that is the first obvious morphological change that we can see, literally see, when we look under the microscope at these tissues when those cells are labeled. The endothelial cells don't look any different when the pericytes are acting that way. You know, they look the same. Now, have we done exhaustive characterization of cell surface proteins and junctional proteins that the endothelial cells are expressing while we see the pericytes doing this? The answer is no, we have not. And that would be something really interesting and important to do. So I tell you the first thing that we see is the pericytes pulling off, but it's quite possible that at the same time, we just haven't looked for it, those endothelial cells could also be freaking out in their own ways.
So do you happen to know the mechanism by which diabetes affects the parasite activity and how that is integrated into the model?
Yeah, it's not integrated into the model because we don't know is the answer. I think that that's something that we need to figure out. I just jumped to the fact that magic insulin, which returns blood sugar levels to normal in those mice, brings the parasites back. But the mechanism by which blood sugar levels and parasite pulling off of vessels is related is a huge question mark. Now, we can hypothesize that it has to do with N-glycation products and other, biochemical changes that are happening as a result of those endothelial cells getting loaded up with sugar, that maybe the response of the parasite is just to not like that, or it interferes with their cell-cell coupling in some ways. But that's the best we can do, is just speculate at this point.
Okay, our next question asks, would it be useful to have 3D cell imaging for analyzing the concentration of TGF-beta?
Absolutely. I would love to measure TGF-beta levels and also all the other inflammatory cytokines in time and space, right? Like it's not just a time problem, but there's also the spatial component as well. So to the extent 3D imaging is possible, that's not something that that we have that much expertise in. Certainly not labeling individual proteins, soluble or matrix-bound proteins, but that kind of information would be incredible for validating our models. I would love to have that at our fingertips and work with somebody if that's something that you're getting.
Okay, so I think probably our last question, hopefully it's from a student. They said, everybody has said to thank you for your amazing talk, but they want to know specifically why you selected parasites as the focus for your work.
Oh, I love that question, especially from a student. So when I was in graduate school here at UVA, this is back in the late 90s, I encountered parasites and I was absolutely captivated by them because nobody really understood what they were. And I say that because then and still now, there was not a unique marker to identify them. And so the only way they've been recognized, even now, is by their morphology, is where they are and what they look like. And that just really intrigued me. And the whole world of angiogenesis, especially back then, was so focused on endothelial cells. The fact that every endothelial cell also has this mystery cell attached to it, which is fascinating to me. And then as I dug in deeper and other people got more excited about parasites too, the fact they perform all these different roles, regulate blood flow in the brain, differentiate potentially into stem cells, instruct leukocytes where to traffic to in an inflamed tissue, and regulate angiogenesis and permeability. Like the laundry list of things that they contribute to is just amazing. And the fact that in disease, they get screwed up, right? Like diabetes, and the fact that that's not happening just in the eye. Everything I mentioned with respect to parasites and these bridging phenomena and vascular dysfunction is also present in the other tissues that we've looked at. So this kind of systemic, they're in every tissue of the body, they're along every capillary, and nobody really knows what they are, was the driving question, and it's still a driving question. So I guess it just piqued my curiosity back then, and because we haven't been able to figure it out, the field hasn't either, they're still interesting to me.
Excellent. So we are surprisingly on time this morning, which is excellent. So we'll give everybody a chance to get to the next session. Shayn, there was quite a few more questions that were on here. So for anybody that didn't get their questions answered, please reach out to her and have a discussion throughout this week. For everybody else that maybe hasn't been on Sococo yet, this is a reminder that just to stay on this Zoom, and there'll be a 15-minute little crash course. And thank you again.
Thank you, everybody. Thanks for your attention.